So we have now arrived at our final destination. In the last two lectures, we are going to talk about this irreversible step of translation. So here we are going to change the molecular language completely. So we are going to go from a linear RNA polymer into a first linear protein polymer, but this polymer will also be able to form fascinating 3D structures and also create function. Here we have again our overview for the flow of information. So we have our double-stranded DNA that's being transcribed into a single-stranded RNA. And basically from here, we have a switched language from a 4-letter alphabet to 20-letter alphabet of the proteins. And today we are going to go through how this language switch occurs at the molecular level. I'm assuming that you are familiar with amino acids, probably already from school. So we have all these different amino acids that are then linked together. And I want to draw your attention to these amino acid groups. So we have on one side the NH2, the amino group, and on the other side the carboxyl group. And the chemistry will be basically linking these one after the other. And the chemistry, as you will see in the next slide, is exceedingly simple. Now we take a look at the basic chemistry of this peptide bond formation. It is actually very easy. Basically, the nitrogen atom will attack the electron deficient carbon on the next amino acid and will form this peptide bond. And the R1, R2 will then specify what type of amino acid this is. And if you then put one after the other, you are going to have the polypeptide chain that then eventually will make your protein. Now to the non-trivial question. On the left, we have our RNA chain that has been formed by a phosphodiester bond, and we have 4 different letters. These somehow get translated into a polypeptide chain with a peptide bond, but 20 different letters. So now the big question is, how does nature accomplish this huge change in molecular language? 2 years later, after their seminal publication of the DNA structure, Francis Crick put forward an adapter hypothesis. So basically, he said there has to be a molecule that ferries around these different types of amino acids, and these have to be then linked together according to the instructions coded on the DNA. He even suggested that there has to be at least 20 different types of these adapters, whatever they are. In 1954, James Watson, together with others, founded this RNA tie club. So on this picture, you see actually Crick on the left and Watson on the right, and you also see them wearing their RNA ties. So in this club, it was Francis Crick who first came up with this adapter idea. And in fact, when you look at the list of scientists that came together in order to solve the riddle of the RNA structure and to understand how it builds proteins, you will find also names that you did not necessarily associate with biology. However, together they came up with brilliant ideas. 2 years after Crick postulated the existence of these adapter molecules in 1957, Hoagland and colleagues indeed came up with the first experimental evidence that such an adapter exists, and this adapter is a soluble ribonucleic acid. And yes, there was a fierce competition to find this soluble ribonucleic acid activity. And the first who has been able to isolate this tRNA was Robert Hawley in 1962. And here in this paper, 1965, they have been able to actually also solve the structure of the first tRNA molecule. In this case, it was the tRNA for the alanine amino acid. And in fact, for this interpretation of the genetic code at the molecular level, Hawley, together with Nierenberg and Khorana, got the Nobel Prize in 1968. So far, we have focused on this translator, the tRNA, as an adapter to basically adapt the languages to each other. But the other question is, of course, how exactly is this genetic code read by the machinery? And this was again a fierce competition. Gold medal in this competition eventually went to Nierenberg and his group. Remember, Nierenberg is one of the recipients of the Nobel Prize in 1968. And when you look at the NIH side, so Nierenberg was the first person to win a Nobel Prize from NIH. So they basically summed everything up to that they have solved with an experiment what others had been unable to solve with theoretical explanations and mathematical models. On the right, you see one of the pages of their lab journals. So basically, in the life science, the experiment will trump everything else if you want to really understand the language of any machinery. Nierenberg and his group single-handedly cracked the complete genetic code with their experiments one by one and put together these 3 letters, these codons that then code for a specific amino acid. On this slide, we see the schematics of this genetic code, how it is basically read from the mRNA, so these so-called codons. Again, important that the translation also has a directionality and it reads from 5 to 3 prime. So this is basically universal. We have 5 to 3 prime replication, 5 to 3 prime transcription, and 5 to 3 prime translation. How does this all work? So let's take a closer look at the translators. So the tRNA on the left, you see the cloverleaf representation of the tRNA. On the right, the 3D structure. So it will have an anticodon loop at the bottom. So this will basically specify which amino acid has to attach to the 3' end. And for all the different amino acids, we are going to have a specific tRNA. So in this case, it's phenylalanine. And these tRNAs will then have to line up one after the other exactly as it has been originally specified in the DNA polymer in order to make the protein polymer. One of the most important steps in protein synthesis is to prepare the correct tRNA. So the example given here is with tryptophan. So we see the tRNA with its tryptophan anticodon, meaning this particular tRNA has to be coupled to the tryptophan amino acid. And this job is done by yet another enzyme, the tRNA synthetase. So this enzyme needs to come in, needs to correctly identify the anticodon for the particular tRNA and connect the correct amino acid. And this machine can also correct its mistakes, meaning it can excise wrongly coupled amino acids and put in the right one. So this again requires energy. So here we need ATP that then creates the high energy bond, if you will. So we are going to take a look at this reaction in detail in the next slide. And basically, when everything went then right, this tRNA will then recognize the tryptophan codon specifically on the mRNA during the protein synthesis. The music plays at the 3' end of the tRNA. So you see this CCA ending. So this is basically a universal signature of all tRNAs, and the coupling of the amino acid will then be catalyzed exactly onto this A. We will need ATP for the activation of the amino acid. So this is going to happen in 2 steps. In the first step, the carboxyl group of the amino acid will attack the first phosphate, the alpha phosphate of the ATP, and will kick out this pyrophosphate. And this pyrophosphate will then be further hydrolyzed into 2 phosphates by the pyruvate phosphatase. And you may actually remember this exact reaction from the DNA polymerization. So what this does is it removes the pyrophosphate from the equilibrium and pushes the product formation here, the aminoacyl adenylate. In the next step, this invariant adenosine at the 3' position of the tRNA will now couple to the aminoacyl adenylate that we have created in the first step. So here again, the 3' OH group will attack the carboxyl group of the amino acid and will create the aminoacyl tRNA that is then ready to go for the polymerization reaction. As mentioned above, some of the tRNA synthetases have proofreading ability. So meaning if they incorrectly couple the amino acid to the tRNA, and this is especially the case for amino acids that look very similar, and in such case, this mistake will be corrected in the editing site, and this step then has as a significant impact on the overall fidelity of the protein synthesis. Finally, we now take a look at the machine that will make these polypeptides. So this is the ribosome. Ribosome is unique. It is an enzyme that combines the rRNA, the ribosomal RNA, with the protein to build this huge machine. So we see here the prokaryotic ribosome. It will then basically dissociate into 2 parts, a large part, so here 50S, and a small part, 30S. So these 2 components, this is not just unique to prokaryotes, but also eukaryotes. So the ribosomes are always made from a large subunit and a small subunit. But then when you look further down, then you are going to see all these rRNAs of various sizes. So at the end, these 2 things come together in order to form this catalytic enzyme to stitch together all these amino acids. On the left, we see the structure of the ribosome, the large subunit that docks onto the small subunit. On the first side, this may look like a protein machinery, but 2/3 of this ribosome is made of rRNA and the rest is protein. And on the right, we see the schematics of this machinery. So you see 3 different sites. So remember, we are going to have a bunch of tRNAs loaded with their amino acids. They need to be basically go through this machine in order to get stitched together. So they enter the machine through this A site, the acyl site, and the next site is the peptidyl site where the synthesis occurs. And once the amino acid is delivered for the protein synthesis, the empty tRNA has to exit the whole machinery, and this then happens in this exit site, the E site. We are going to go through this translation process step by step. First, we are going to look at simplified schematics, but the concept of the translation is basically the same as we also saw with the DNA polymerase or RNA polymerase. We are going to need an initiation step, and then the polymer needs to elongate, and eventually we are going to need a termination step. In our previous lectures, we saw that the replication machinery or the transcription machinery can recognize really complicated words. So here, in the case of the translation machinery, it's actually quite easy. So this methionine, if you will, the word now, so this is the codon AUG, is basically the universal start for all proteins. And the stop sign is also, again, a codon. In this case, we have 3 different codons. And once the machinery walks by this particular codon, the synthesis will be terminated. So we have to find the methionine first on the mRNA. So how this happens, again, different for prokaryotes and eukaryotes, but we are going to go into these details later. So here we are going to have the small subunit. Remember, this was the site where the codon, anticodon matching occurs. So as soon as this methionine is found, the large subunit comes in, assembles the complete machinery, and everything is now ready to go. As we have seen with the other machines, that we need always some factors in order to initiate something, elongate something, terminate something. And here we have the EFTU as the elongation factor for the ribosome. So this then brings in the next tRNA. So you see that this enzyme is now loaded with the GTP and inserts the tRNA into the A site. Once this new tRNA is inserted, we are going to kick out the old tRNA. So here, in this case, the yellow tRNA is now kicked out and the exit site is now freed, number 1. Number 2, we are going to have 2 outcomes with this newly incoming tRNA. Either it's going to be incorrect, so the codon anticodon matching will not work. So this one will preferentially dissociate. So this is basically the proofreading step. Or the tRNA, the new tRNA is the correct one. Then the elongation factor hydrolyzes GTP to GDP and cock this new tRNA into position for the next peptide bond formation. Because the blue tRNA was the correct one, we now have this chain elongation, but now we have to switch places. So the old tRNA, the purple one, has to get into the exit site to be kicked out, and the blue tRNA has to prepare for the next round of peptide formation. So the A site needs to be freed. And this all happens through huge conformational changes that is then coupled to yet another elongation factor, the EFG. After the GTP hydrolysis, this elongation factor will release from the A site, so free the A site for the next tRNA to come in. And now we have placed the blue tRNA into the P site to wait for the next peptide bond formation. Eventually, the machinery will pass by a stop codon. And once this stop codon is in the A site, so this is the sign for the release factor to come in and block this site. And this is the sign then for the machinery to release the polypeptide chain and to disassemble. One thing to note is that the stop sign does not have any tRNA, right? So the start sign had its tRNA, the methionine, but we do not have any tRNAs specifically to recognize these stop signs. In the next slide, I'm going to show you the visualization of this elongation step so that you get a better idea of how these steps basically are coupled one after the other.
To extend a growing polypeptide chain, the ribosome must select the correct amino acids that are specified by the messenger RNA. An aminoacyl tRNA bound to elongation factor TU, EFTU for short, enters the free A site on the ribosome. If the anticodon of the charged tRNA does not match the codon in the messenger RNA, the tRNA is rejected. The process of trial and error repeats until the correct tRNA is identified. Elongation factor TU hydrolyzes its bound GTP and dissociates. If the tRNA is correctly matched and remains bound for a long enough time, it is committed to be used in protein synthesis. The ribosome catalyzes the formation of the new peptide bond and undergoes a dramatic conformational change. Elongation factor G binds to the ribosome. Hydrolysis of GTP by elongation factor G switches the ribosome back to the state in which it can accept the next incoming tRNA.
So we have just went through the overly simplified translation process. And as you can imagine, this process is, of course, way more complicated than the way we looked at it. So this huge machinery has to basically orchestrate all these events that take place at different locations, like the codon, anticodon matching in the small subunit. This information has to basically pass on to the large subunit in order to make the peptide synthesis. And we are going to also take a look at these reactions in a little bit more detail in the next lecture. Before we conclude the first part of the translation process, we then ask our famous question. So how does it benefit me? This knowledge of knowing how this translation machinery actually works. Well, you remember the penicillin, the first antibiotic that has been discovered by Fleming. So you probably also heard about people who are allergic against antibiotics, some types of antibiotics. And it turns out, indeed, that we can also develop antibiotics against the translation machinery of the E. coli, for instance, that is different enough from our translation machinery that we can specifically block the march of the large subunit or block specifically the A site or even block the elongation factors. And knowing the mechanisms will get us, of course, again, far in terms of diversifying the tools that we have in our hands in order to fight diseases. In the next lecture, we are going to go into a little bit more detail of the translation. So we are going to make the distinction between the prokaryotic and the eukaryotic translation machinery. And we are also going to learn a little bit more about how these reactions are actually catalyzed. So as always, if you have any questions to part 7a, please drop me a line.